Hello and welcome to this GCSE Science Revision video for additional biology on organisms and the environment. In this video we will look at photosynthesis and the structure of the leaf, limiting factors, how plants use glucose and how we measure the distribution of organisms. Green plants absorb light energy from the sun in chlorophyll in their leaves. They use the red part of the light. That is why chlorophyll is green in colour. Within the leaf, or within the leaf cells, the photosynthesis reaction takes place. This converts carbon dioxide from the air and water from the ground into glucose, which the plant uses, and oxygen, which is given out as a byproduct. Many ways can be used to measure the rate of photosynthesis. However, in your exam, they may give you an experiment looking at pondweed. Pondweed is a type of plant which is grown underwater and gives off bubbles of oxygen as it photosynthesizes. They may ask you to comment on the amount of bubbles of oxygen that are given off under different conditions. The more oxygen that is produced, the greater the rate of photosynthesis. Photosynthesis is dependent on the amount of light, or the amount of light that's absorbed. It's also dependent on the amount of carbon dioxide in the air, the amount of water in the ground, and the temperature that photosynthesis is happening at. Photosynthesis all centers around the structure of the leaf. At the top of the leaf is an upper epidermal layer. The upper epidermal layer, with a thin waxy cuticle on top of it, is very thin to allow light through easily. Underneath that is the palisade layer. The palisade cells have lots of chloroplasts in them, as this is where most of our photosynthesis will be taking place. Underneath that is a layer made up of spongy mesophyll cells. The spongy mesophyll cells, as they are rounded, cause large air spaces to form. This allows carbon dioxide to diffuse into the leaf and oxygen to diffuse out. It's not shown on the diagram, but there are veins in the plant with phloem cells and xylem cells. The phloem cells carry away glucose made in photosynthesis, and the xylem cells carry water to the palisade cells to be used in photosynthesis. The equation for photosynthesis is carbon dioxide plus water goes to form glucose plus oxygen. If you are doing the higher paper, you may need to know the chemical equation for this. The chemical equation is 6CO2 plus 6H2O goes to form C6H12O6, which is glucose, plus 6O2. When describing photosynthesis in relationship to the leaf, there are a number of stages that it needs to be split up into. In stage 1, you need to talk about the light enters the upper part of the leaf. The part of the leaf, so the chlorophyll, absorbs the red part of the spectrum. Chloroplasts in the palisade cells absorb the light energy. Water enters the leaf through the xylem and leaves through the stomata by the process of transpiration. Carbon dioxide diffuses into the leaf through the stomata. Carbon dioxide and water are both needed for photosynthesis. They are called the raw materials. Both water and carbon dioxide diffuse into the palisade cells. So therefore, photosynthesis occurs within them. Oxygen made during photosynthesis therefore diffuses down a concentration gradient into the air spaces and out of the leaf. And finally, the glucose made in photosynthesis is transported to other parts of the plant through the phloem. When considering photosynthesis, we need to consider a few limiting factors. A limiting factor is any factor that will slow down or limit the amount of photosynthesis that a leaf can carry out. The first limiting factor is light intensity. Without enough light, a plant cannot photosynthesize very quickly, even if there are plenty of water and carbon dioxide. So increasing light intensity will speed up the rate of photosynthesis. Sometimes photosynthesis is limited by the amount of carbon dioxide in the air. This does not happen very often, however. So even if there is plenty of light, the plant can no longer photosynthesize any faster if there is insufficient carbon dioxide. The rate of photosynthesis is affected greatly by the temperature. If it gets too cold, then the enzymes carrying out photosynthesis in the chlorophyll don't work fast enough. If it gets too hot, then these enzymes can become denatured, so no longer work. The other main type of limiting factor might be the amount of chlorophyll within the leaf, as if all the chlorophyll is in use, then the rate of photosynthesis cannot get any higher. Plants use the glucose that they make during photosynthesis in a variety of ways. The plant may store the glucose in insoluble substances such as oils, fats or starch. These substances can then be used later on if the plant needs them when it is not able to photosynthesize. Glucose and starch can be converted into other substances by the plant. So, for instance, they can be converted into cellulose for cell walls or proteins for growth and repair. The advantages for the plant of using an insoluble substance such as starch for storage rather than soluble substances such as glucose are that they do not affect the water concentration inside cells and they cannot be washed away. The final section in this that we need to consider is how we measure the distribution of organisms. It is not usually possible to count the entire population of a particular organism in a habitat. This means that the population must be sampled. The two main ways that this is done is by the use of transects and the use of quadrats. A transect is a line across a habitat or a part of the habitat. It can be as simple as a string or rope being placed on the ground. The number of organisms of each species can be observed and recorded at regular intervals along the transect. Quadrats can be used to measure the number of organisms within a specific area. Quadrats are usually square, and they might be 5 by 5 or 10 by 10. The quadrat needs to be placed randomly within an area and the organisms need to be counted in it. To make the results more valid, a number of different areas need to be sampled using the quadrat and then the results need to be averaged out. A different type of transect that can be used is a belt transect. In a belt transect, belts across a particular habitat are marked out and then all the organisms within a particular belt or within particular areas within a belt are counted. With belt transects, we can go and not only sample the different types of organisms within an environment, we can also sample soil type, or pH of soil, light intensity, or the amount of water. So remember, when revising, start earlier rather than later. Keep questioning yourself using flashcards. Quality of revision is better than quantity of revision. Good luck with your exam.